Hi, everybody, and welcome once again to our online lectures. In this opportunity, we are going to talk about the parts of the cell, or how we like to call them, the organelles of the cells specifically. So let's start by talking about why are cells so small, if you have ever wondered. The size of the cell, it's influenced by the service that it provides. Large cells, what we can say about them is that they have a huge surface area. And smaller cells have a larger surface area in relation to their size. And what is all this surface area thing that we're talking about? Think about it as space to do work. Even though a large cell might have a huge surface area due to the size of the cell, but by having multiple cells that are smaller, closer together to one another, they have more space to do more work. So, in other words, in the same space that you can fit a big cell, if you can fit a bunch of smaller little cells, they can do 3, 4, 5 times the job of a cell that it's really, really, really big, making them extremely efficient. And efficiency, it's really pretty much what we want when talking about biological components. And all of those functions, all of that functionality that occurs in cells, it all occurs across the plasma membrane. And if you remember from our previous video, all of the cellular transport, the passive diffusion, the diffusion, simple diffusion, facilitated diffusion, osmosis, all of that occurs through a plasma membrane. So if I could do more of this with less size, it's going to be really, really, really good. So that is what we call the benefits of being really small. Let's do a little refresher on the plasma membrane because of the importance of the plasma membrane for living things. The plasma membrane, or a synonym for the plasma membrane, would be the cell membrane. It's an interchangeable concept. It's a flexible boundary between the living cell and the unliving surroundings of the cell. It separates the outside from the inside. The main component of the cellular membrane, it's the phospholipid, and as we discussed before, there is two main regions to the phospholipids. We have the head, or the phosphate group, and we have the tails, or the fatty acids. And the important thing about them, it's the concept of hydrophilic and hydrophobic, meaning hydrophilic means water loving and hydrophilic means water fearing. And it is exactly by having those two very particular abilities in the same molecule what constructs the plasma membrane the way that it is and gives it the function that it really, really, really have. So talking a little bit about specific about the what is regulated. So the flow of materials are regulated by the plasma membrane, and molecules, for example, like carbon dioxide and oxygen, have the ability to move freely across the plasma membrane, or in other words, using simple diffusion. And if CO2 and oxygen kind of like ring a bell, that it's exactly the gases that we exchange via our respiratory system, and we do this constantly. So having the ability to do this really fast across any part of the plasma membrane, it's definitely an advantage to have. Other materials like salts and water required special proteins to be able to pass through the membrane, and that's what we discussed as the process of facilitated diffusion. So one important thing about cells, and I kind of make a little joke there, and I put an old cell phone there. One thing that we should know is that there is two different kinds of cells. Just like I mentioned before in previous videos, we have eukaryotic cells and we also have prokaryotic cells. And the big difference between these two types of cells is the presence or absence of the nucleus of the cell. Eukaryotic cells are said to have a membrane bound nucleus, or they have a very well defined nucleus, and prokaryotic cells do not have a membrane bound nucleus. As a matter of fact, the region where the DNA is located inside of a prokaryotic cell, like a bacteria, it's called a nucleoid, or a false nucleus. So I think that it's very important to keep those things in mind, and also keep in mind what is our type of cell, what is the type of cell that you can find in human beings, and that would be eukaryotic cells. All right, so very important to distinguish between those two things. So here we just have some information about what prokaryotic cells are. We already said the important things about them, which is that they do not have a membrane-bound nucleus. So we're just going to move forward with those bacteria and concentrate on the cells that we can find in human beings. Like I said before, the main feature of eukaryotic cells is the presence of a nucleus. Inside the cell, we have their little parts or little organs, and they go by the name of organelles, right? They are by the name of organelle. Each of these organelles, it's bounded by a lipid protein membrane. So the organelles inside of the membrane, just like the cell itself, are surrounded by a membrane as well, according to what compartmentability was when we talked in chapter 1 about the requirements of life. We can divide these organelles in different groups according to their specific functions, and those functions are the following. Some of them are in charge of genetic control. Other ones are the makers, or they are in charge of manufacturing and distributing and breaking down molecules. Other ones are there for providing energy for the cell. And then we have others that are specifically there for offering structural support or maintaining the size and shapes of different cells. This is just to do a little contrast between one cell and the other. This is the example of a plant cell. And I just wanted you to see that even though plants are completely different from, for example, a human being or a person, their cells follow a lot of similarities, like having a nucleus and having those membrane-bound organelles and many other things. But our main focus, of course, is going to be about the eukaryotic cells, specifically animal cells, like the cells in our body. So I think it's time now to start talking in detail about each of these organelles of the cell, learn how to recognize them from a diagram, and obviously, and of course, their main functionality. So let's start with the most important one, or the most well-known organelle of animal cells, and that would be the nucleus. The nucleus contains the genetic information of the cell. So it is in here where you're going to find the instructions, or the cell. It's going to find the instructions for doing every single cellular process in a living organism. Inside of the nucleus, the DNA, it's organized in a structure called a chromosome, right? And in the case of humans, I would like you to put that out there. In the case of human, we have 23 pairs of these chromosomes, which would give us a total of 46 chromosomes. And that number is what defines a human as actually being a human. Sometimes in textbooks, you're going to hear the word chromatin, and it sounds really close to what chromosomes are. The big difference is that chromatin, it's what we call an uncondensed form of DNA. Chromosomes are only visible when the cell is about to divide from one cell to two cells, but during their normal functionality, the DNA, it's an uncoiled kind of configuration that we call chromatin. So the difference between chromosomes and chromatin, it's pretty much their configuration. Condensed, they're going to be chromosomes. Uncondensed, they are going to be called chromatin. The nucleus, it's enclosed in a double membrane that has a specific name, and that's called the nuclear envelope. And inside of this nucleus, besides of the genetic material, the DNA, there is another structure there called the nucleolus. And the function of this secondary organelle inside of the nucleus, it's to synthesize or create what we call RNA, that eventually we will learn a lot more about that specific RNA. This is what it's used for creating proteins later on cellular processes. Another organelle, the ribosome. Ribosomes are also very important as well because they carry a specific function of building proteins, right? They are completely in charge of building proteins. They build proteins based on information that they can get from a molecule called RNA produced inside of the nucleus in there. Um, and those ribosomes will use that information to make me proteins. And I can find them in two different places. in animal cells. They could be free, floating around the cytoplasm. And let's say what that cytoplasm is all about. Cytoplasm, it's the space between the membrane of the cell on the outside and the membrane of the nucleus. That is what the cytoplasm is. It's pretty much all of the space inside the cell except for what's inside of the nucleus. So we can find them free, floating around, doing their thing, doing their proteins, or I can find them attached to another organelle that we'll discuss in a second called the endoplasmic reticulum. The free ribosomes build proteins used in the cytosol, which is the liquid substance in the cytoplasm, and the attached ribosomes build proteins that eventually later on will travel to different parts of the cell, either to be excreted outside the cell or to be modified to a specific function. So ribosomes are very important due to the fact that they build proteins. And proteins are fundamental little things necessary for life to happen in organisms like human. Now let's talk about the endomembrane system, which is a series of compartments that I can find in eukaryotic cells. The components of the endomembrane system, there are several organelles that fall onto this system. We're going to mention them now, and then we are going to talk a little bit about their specific functions. So we have the nuclear envelope, the endoplasmic reticulum. We have another organelle called the Golgi apparatus. We got lysosomes, we got vesicles and vacuoles, and of course the plasma membrane in itself. The endoplasmic reticulum specifically, for short we call it ER, it's a network of flattened sacs and little tubules that are spread all around the cell. The ER is a continuous membrane from the nuclear envelope. That means that they're built of the same thing, and it divides into different compartments that allow that allow different functions to occur in there at the same time. Talking specifically about the endoplasmic reticulum, there is two different types of it. We have the smooth endoplasmic reticulum, and we have the rough endoplasmic reticulum, and their structural difference, at least, is the presence or absence of ribosomes attached to the endoplasmic reticulum. In the smooth, there is no ribosomes attached, and in the rough endoplasmic reticulum, there are ribosomes attached to this. So that would be a structural difference between the two of them. Now let's talk about a difference in their functionality. The smooth endoplasmic reticulum, it's there for the synthesis or the making of lipids. And remember that when we said lipids, we are talking about fats. And some kinds of fats that could be created in the smooth endoplasmic reticulum are oils, phospholipids, and steroids as well. If we talk about the rough endoplasmic reticulum, then this would be very important in the production of proteins as well. And we can just see this because of the presence of the ribosomes attached to the rough endoplasmic reticulum. As an example, insulin, which is produced by the pancreas, it's produced in the rough endoplasmic reticulum. And after it's created there, one of those functions of the ER is that it can create vesicles that can be used for transferring their products to other places. So you can think about it as a little factory, and then it can wrap up whatever they produce, and they can ship them away to some other place that might be needed. For example, insulin, it's used in the blood for lowering or metabolizing sugar. So imagine this being created in the rough endoplasmic reticulum, being packaged in a bubble made of the same thing of the plasma membrane, and that little bubble can exit the cell and it can travel to other places. So that would be the main difference between these two organelles, smooth and rough ER. The first difference would be, of course, the ribosomes in there or not, and the other one is their functionality. Smooth, it's for fats. Rough is for proteins. Talking about their packaging, this is just, I want you to observe this picture here to have an idea on how this packaging actually works in the rough endoplasmic reticulum. So you can see a ribosome creating a protein, creating a polypeptide. And then inside of the ER, it could be customized. We can add things to that protein, and then that protein kind of like travels to the outside or to the edge of the endoplasmic reticulum, and from there it forms this little bubble, which we call a secretory protein. So it can travel along the cytoplasm of the cell or the cytosol of the cell, and then it can exit and it can go someplace else. So that's the functionality of this endoplasmic reticulum. Let's talk now about the Golgi apparatus, which is similar in appearance to the endoplasmic reticulums, but has a very different functions. A bit very different function. The Golgi apparatus serves as a molecular warehouse and a processing station. Pretty much what it does, it removes or modifies sugars that are attached to the proteins that were once created for the endoplasmic reticulum. So you pretty much can think of the Golgi apparatus as a customization organelle to give proteins a more specific functions, to tailor the proteins to a very specific functionality. So all of these organelles will work together one with the other, creating whatever the body needs. So you can think about it as the ribosomes are the rough workers that create the protein, and the ER will customize it, package it up, and then in case that there needs to be any additional packaging that we need to do, then that's what the Golgi apparatus is there for. So a good difference to know about the Golgi apparatus is that the main function of this organelle is not to synthesize anything. It's to modify the products of other organelles, specifically the endoplasmic reticulum. Lysosomes are what we call the recycling center, right? This is the recycling center of the cell. The lysosome, it's a membrane enclosed little sac of digestive enzymes. And digestion always, we can always associate the word digestion with breaking things down. The enzymes and membrane are built by the endoplasmic reticulum and the Golgi apparatus, so they kind of like are in charge of pretty much creating the lysosomes, and like we said, they are my recycling centers. Why do we need recycling centers? Simply because the materials of anything inside the cell could be reused by the same cell, and I don't have to acquire those materials from anywhere else, right? What kind of materials can be broken down in there? For example, defective organelles could be eaten by lysosomes, broken down into their individual components, and they're released into the cell so that I can make me more. So in other words, I can recycle the organic material inside the cell thanks to the function of those lysosomes. Talking about vacuoles, vacuoles are like big bubbles inside the cell, and they are pretty much there for storing different kinds of things. Plant cells would have vacuoles, animal cells would have them as well. But in animal cells, the vacuoles are technically smaller, and they are in charge of storing pretty much a little bit of water, or they can storage electrolytes for later use. And that's pretty much what we should know about our vacuoles. They're just little storage centers. You can think about them as little chests that will carry different materials, right? This is the exact same vacuole that we were talking about when discussing the endoplasmic reticulum. So now this organelle requires a little bit more of a nice attention to it. The mighty mitochondria. This is one of the most well recognized organelles in eukaryotic cells, simply because they are the batteries of the cell. They are the powerhouse of the cells. Inside of this mitochondria occurs a process called cellular respiration, which cellular respiration pretty much involved using oxygen, transforming this along a big metabolic process into carbon dioxide, and using oxygen to create ATP, which is the molecule of energy. Now, that name right there, ATP, very important once again, very important. This is the currency for energy. This is the molecule that every single process that will require energy, this is the molecule that they would use, right? This would be the mitochondria. The mitochondria itself, it's enclosed in a double membrane made from phospholipids. So it's pretty much the same material of what the cellular membrane on the outside it's made for. And looking at the inside of a mitochondria, we have two parts that are considerable. One of them is the matrix, and the other one is the crista. The matrix will contain mitochondrial DNA and ribosomes that direct the cell respiration. So if that strikes you as a little weird, yes, the mitochondria possesses DNA as well. Possesses DNA as well. And inside the matrix and in the crista, it's where the actual respiration occurs, the transformation of oxygen into carbon dioxide. This is where the sugars that we ingest during the day would eventually go to be broken down in the presence of oxygen to produce that molecule, right? To produce the ATP molecule that we talk so much about in every class that has to do with biology. The chloroplasts are just in here because we are talking about cells. This is an organelle that is exclusive to plant cells, and it's not necessary to study of them specifically for our class at this time. I like to talk a little bit about, just to put some context, some background about this weird organelle that has DNA. It's the only organelle besides the nucleus that will contain some organelle. And talking about symbionts, this is pretty much a theory that was proposed many years ago that the bacteria and chloroplast, at a long time ago, they were organisms by themselves, right? They were like eukaryotic, like prokaryotic organisms, like bacteria. And at some point, they got introduced inside of a cell and they started doing a symbiotic relationship, one with the other. And well, the mitochondria could benefit from the materials in the cell, and the cell, of course, would benefit from the materials produced by the mitochondria, which is ATP, and that's what the cell uses for its energy. Okay, so that was just a brief discussion on our organelles, on those little parts inside of the cell, and by understanding how they work, how they look like, what is their functionality, we can understand a little bit later on how is it that cells can work together one with the other to move to the next level of organization, which is of course the tissues, and have a better idea on their functionality, or where does the dysfunctions will actually come from. All right, well, thank you for your attention, and I will see you on the next video in which we will be discussing the four fundamental tissues of the human body. Thank you, and see you on the next one.